Acid-base indicators are chemical dyes or stains that exhibit specific color reactions with acids and bases. There are quite a number of commonly used acid-base indicators, each with their own unique color-changing reactions towards acids and bases. Among these, litmus indicator, phenolphthalein, and methyl orange are probably the most commonly encountered indicators. In this simple yet visually satisfying experiment, we shall make a side-by-side comparison of how different acid-base indicators show striking color changes when exposed to an acid and a base sample. For reference, we shall also include a pH 7 neutral solution to see how they behave color-wise in such a neutral solution. To perform this experiment, we first gathered together an assortment of commonly used acid-base indicators. For this video, I've poured out samples of each indicator into clean and dry test tubes to have a visual overview of their original colors in solution. Some of these indicators are water-soluble dyes, while the rest are soluble mostly in alcohol. Accordingly, their respective solutions were prepared using either water or alcohol as a solvent. Next, we arrange 3-row cell culture plates or microplates with as many number of wells as the number of indicators to be tested. We label the 3 rows as acid, neutral, and base. We also label the columns with the names of the indicators to be tested as shown here. The 3 rows are filled with few milliliters of a dilute acid, a pH 7 buffer solution, and a dilute base respectively. For a better visual effect, I have used cell culture plates. You may also use several test tubes instead. Next, using separate clean droppers for each indicator solution, we add a few to several drops of the indicators to the wells marked acid, neutral, and base, following the labeling pattern of the indicators. Once all the indicators have been added to their designated wells or tubes, we can clearly observe the diversity in the color reactions of each indicator with acid, neutral buffer, and base. This concise table shows a list of the acid-base indicators that we have tested, their effective pH range, and their color change patterns in an acid and a base. Compare the colors obtained with this table. One interesting observation we may make from the experiment is that some indicators such as thymol blue show unique color changes not only in an acid and a base solution but also in the neutral buffer solution with a pH of 7. Similarly, in the case of bromothymol blue, you can see 3 different colors in acid, neutral, and base solutions. In the case of bromothymol blue, the neutral pH 7 happens to be within the transition range of the indicator and therefore shows a mix of yellow and blue, which is green. The pH transition range of an indicator is the pH range within which the 2 colors above and below that range sort of mix to give a different color. This is the range where the color on one side transitions into the color on the other side of that range. This transition is visually characterized by a mixing of the 2 colors above and below that range. This behavior of certain indicators can be correlated with their effective pH range and their pH transition range as shown in the table. This makes them a versatile and dynamic indicator agent to detect minute changes in pH in a chemical reaction. The universal indicator solution that you have probably come across very often in the lab is in fact a complex mixture of several acid-base indicators with varying effective pH ranges and shades of color at different pH levels.